Phytobiome. This refers to the entire system of factors that affect or are affected by plants. It consists of the plants themselves, the environment, and their associated communities of microorganisms. This is microbial communities, the soils and plants themselves, insects, nematodes, or any other animals that are that are associated with plants, as well as their environment. So understanding the phytobiomes is important for enabling sustainable and profitable crop production while minimizing negative impacts in the environment. So it's very closely associated with some of the things we do with soil health. And of course, soil health is very closely associated with plant disease management. By definition, healthy soils will have less disease than a less healthy soil. We know that soil-borne diseases tend to be most severe when soil conditions are poor, when you have some factors like inadequate drainage or poor soil structure, low organic matter, fertility, high soil compaction, low microbial biomass. All these things can contribute to more susceptibility to disease and higher disease problems. So most practices that will improve soil health will also reduce soil-borne diseases. By improving those conditions for crop growth, you can reduce disease by increasing that microbial biomass and activity. That increases general disease suppression and also potentially increase populations of antagonists. However, in many cases, increasing or improving soil health in general may not control soil-borne diseases enough, or at least to the level that we need for crop production. So there's further things that need to be done, or that other more specific management practices that need to be taken to improve this reduction of soil-borne diseases. And that gets us into the topic of disease-suppressive soils. And these are soils in which the disease is not readily developed, despite the presence of both the pathogen and the host. These can be due to physical or chemical factors, but most often it's a result of soil microbiology, the activity of specific soil microorganisms or whole microbial communities. And these disease-suppressive soils can be induced or developed through changes in those soil microbial community characteristics. So if we look at some of the management practices that have the biggest effect or most affect soil microorganisms, they are a lot of the things that are associated with what we talk about when we're improving soil health. The main crop itself is important for influencing soil microorganisms, from the cultivar used, its level of resistance to diseases, the root exudates it produces, and its growth patterns and type of root growth, and other planting factors are all important. Other plants in the system, such as your rotation crops or cover crops, green manures, or any other plants in the system, from crop type to rotation length, rotation sequence, cover crops, other amendments, or whether they're organic, chemical, or biological, all can be factors, and other physical things like tillage and irrigation. As you can see, these are all things that we talk about for management practices for soil health. But today I'm focusing on the plant-related aspects, the crop rotations, cover crops, and the reason for that is that plants are the primary drivers of change in soil microbial communities. Microbes respond to the abundance and diversity of the organic root exudates, which affects the microbial density, activity, structure, composition, and function. So a greater diversity of plant types or plant species will yield greater microbial diversity and activity. And on the flip side of that, specific plant species and genotypes may select for specific types of microorganisms with varying characteristics. So we can use plants, actively growing plants, to alter, manipulate, and even manage microbial communities for better crop growth and reduced disease. And this is what we refer to as active management of soil microbial communities. And it has great potential for being able to harness this for developing disease-suppressive soils and increasing plant productivity. The major limitation at this point is we just do not know enough about these specific relationships and interactions among the plants and soil microorganisms to be able to fully implement this. We are learning more and more all the time now that We have much better tools available now with the advanced molecular techniques and next-gen sequencing. Now we're able to look more specifically at what's going on within the soil and these interactions. But much more research needs to be done to get to that point. A few examples of how this works. First, looking at effective crop genotype. This is work that I did many years ago where we had a specific, a unique cultivar-specific induction of a disease-suppressive soil, in which growing a particular watermelon cultivar resulted in disease suppression. And so on the chart here on the left, under normal situations, when you have a normal susceptible crop such as Florida Giant in the blue bars, when you plant multiple plantings of that over time, pathogen populations increase dramatically. And so you get high levels of disease with devastating losses. When you plant the cultivar Crimson Sweet, however, you have very small change in, which is the red bars, very small change in pathogen populations and lower disease. On the other side of that, if you look at populations of other Fusarium Foxysporum, basically the non-pathogenic strains, with the Florida Giant variety, you get little change, but with the Crimson Sweet, we're increasing those non-pathogenic strains. And so this relationship between the pathogen and non-pathogen is responsible for the disease suppression. But just by changing the cultivar, it completely changed this relationship of pathogen and non-pathogen. As an example of more general effects due to rotation crops and cover crops, this is work that we did looking at different rotation crops of potato with and without a cover crop of winter rye. This is effects on black scurf of potato, which is caused by Rhizoctonia solani, a soil-borne disease. And the crops we looked for as rotation crops here are barley, canola, green bean, rapeseed, sweet corn, soybean, and no rotation or continuous potato. If you look at the back row, which is no cover crop added, you can see there's some variation with the different rotation crops. Soybean and potato, non-rotation has highest level of disease, and the other ones are a little bit lower, with the lowest being canola and rapeseed in the green and purple bars. You compare that with the front row, which is with a cover crop, and you can see it pretty much across the board there, adding that fall cover crop of winter rye decreased these disease levels by about 10 to 20%. So in addition to the effect of rotation crop, you have, by adding the cover crop, you've increased your crop diversity, and you've increased the amount of time that you have active living plants in the system, which are two of the primary principles of soil health. Then you have the combination of that, the combination of a good rotation crop with the cover crop, and you've reduced the disease in this case by 40 to 50%. And this brings us to the use of disease-suppressive crops. Disease-suppressive crops are those that have specific active mechanisms that result in disease suppression. In the previous example, canola and rapeseed are both brassica crops, and brassica is a particular group of plants that have specific properties that make them suppressive to a number of soil-borne diseases, as well as nematodes and some other things. Sudan grass and its related plants also have disease-suppressive properties. But for both of these groups, the disease suppression is a result of multiple mechanisms of action. You have disease suppression due to what's referred to as biofumigation, which is these plants produce compounds that, when broken down, release volatile toxic metabolites, which can kill plant pathogens and suppress these diseases. And also, in addition to this, and just as important, and maybe even more important, is that the disease suppression is due to changes in the soil microbial communities. And these changes are partly independent of their biofumigation properties. But because these types of plants tend to have this biofumigation activity, they tend to be most effective when used as green manure, so that those breakdown products can be used for that. So as examples of how these disease-suppressive crops can be used in cropping systems, I want to talk a little bit about some cropping system trial we did several years ago. And this is where we tried to set up 3-year cropping systems. This is again in the potato system. 3-year cropping systems that were set up to focus on specific management goals of soil conservation, soil improvement, and disease suppression. So we took our standard 2-year rotation, which is the norm in potato production, at least in the Northeast where I'm from, and modified that to get more soil conserving principles. So our soil conserving system, we had a small grain forage grass that was done entirely no-till in the non-potato year, with the only tillage done in the potato year. We also added a straw mulch in the fall after potato harvest to reduce erosion there, to implement a number of soil conservation principles. Our soil improving system started with this same system, but now we added large yearly compost amendments to increase organic matter and kind of improve those soil qualities. In our disease-suppressive system, we tried to maximize the use of disease-suppressive crops with cover crops. So we had disease-suppressive green manures with different cover crops each year, and compared that with the standard rotation and that. And this we did for a number of years. I don't have time to go in details of all the things that we learned in this study. The cropping system did significantly affect the soil physical, chemical, and biological properties. All rotations increased a lot of the factors such as aggregate stability, water availability, microbial activity. The soil improving system, where we added the compost amendment, not surprisingly, had the greatest effect on most of these soil health parameters, increasing organic matter and nutrient content, microbial biomass and activity, improving soil structure, water availability, and all that. But what I wanted to get to is just illustrate the overall effect of these cropping systems. This shows effects on with and without irrigation on total tuber yield. If you look at the front row of bars, that's non-irrigated, and you see the blue bar on the left side is the soil improving system, which had the highest yields, over 40% greater than the non-rotation, and over 30% greater than the status quo or the standard rotation. The disease-suppressive system had the next best. And this also illustrates the kind of increased water holding of the soil improving system. Under irrigated conditions, the back row of bars, you see that pretty much all of them increase substantially with irrigation. But in this case, the disease-suppressive system actually averaged higher yields than any other system. And there was not much difference in the soil improving system between irrigated and non-irrigated, indicating that increased water holding capacity. But looking at this in aspect to the disease reduction, again, non-irrigated is the front row of bars, and you see the green bar, the disease-suppressive system had much lower disease. Again, this is black scurf, Rhizoctonia solani. All the rotations reduced disease relative to no rotation, but the disease-suppressive was lower than any other. We're reducing disease by over 40% compared to the standard rotation. But notice that the soil improving system here, even though it had much better soil quality and soil health parameters, the disease reduction was not any better than the status quo system. It was better than no rotation, but not any better than the status quo system. And this shows too that in many cases, just improving soil health, using a lot of standard measurements, you may not get the kind of disease levels that you need for good crop production. So that's why the use of these disease-suppressive crops can really enhance this level of disease reduction. And this is also observed with the second disease here, common scab. I had first one was black scurf. This is common scab. And to give an idea of the effects on soil microbial communities, this is just a representation of fatty acid FAME profiles, which gives an indication of relative differences in microbial composition. And you see that each cropping system has a distinct characteristic profile that distinct from the others. Up in the upper right section there is no rotation. At the extreme left is the soil improving system, dramatically different from the other rotations, that showing those effects of those increased organic matter and actions. At the bottom right is the disease-suppressive system. And this one, you can see how both the soil improving system and the disease-suppressive system had dramatic effects on soil microbial communities, but very different effects. In the disease-suppressive system, tended to have higher fungal levels, higher actinomycetes. In the soil improving system, we had more mycorrhizae and various other characteristics, but that particular characteristic didn't result in very high disease suppression, whereas the disease suppressive did. So this just gives an indication of how these kind of things affect soil microbial communities and how these can be used to reduce these diseases and develop disease-suppressive soils. I just wanted to make a quick note regarding the organic matter effects on soil-borne diseases. As you saw in the soil improving system, we did get some reduction of disease, and we know that overall increasing carbon and organic matter will result in increased microbial biomass and activity and some disease suppression, but in practice, the results can be highly variable. Various surveys in the literature have looked at this, and so like in evaluations of a wide range of composts, one group found that a little bit more than half of them resulted in disease suppression. Most of the rest had no effect, and a small percentage actually increased disease. Another survey that looked at all kinds of different organic amendments found similar results in that about half of them would result in disease suppression, and a smaller number actually had some increases in diseases. So these kind of things, organic matter effects, can be highly variable, and so just by increasing organic matter, you may or may not reduce disease to the levels that needed. So that's why some of these other approaches are needed. So in conclusion, then, you can look at some of the things here that I've talked about. Incorporating these management practices that promote soil health does result in enhanced nutrition yield and disease suppression. The use of actively growing plants as their main rotation and other cover crops can be used to effectively to affect these soil microbial communities, to increase this antagonism towards the pathogen. And use of specific plants with disease-suppressive properties can greatly enhance disease reduction and development of disease-suppressive soils. So active management of soil microbial communities for enhanced crop productivity and disease suppression is feasible. It can produce very good results, but we do need more research to be able to fully implement to the level. So I hope just with this talk, it's given you some indication of the kind of things that the importance of that actively growing plants in your system and how they can affect and change microbial communities, and how this can be used to develop disease-suppressive soils. And I'll stop there. If there's time for questions, I'll be happy to take those.